Hi guys, welcome back. So just for the interest of posterity and those tuning into this crowdcast recording who weren't here 15 minutes ago, this is session 2 of our Neuroimaging Connectomics morning session. And where we left off back at the end of the last tutorial was just we're going to finish up with our diffusion MRI and diffusion MRI tractography Connectomics practical session, and then we have 2 more for you, which is Fmri and EEG MEG. So I'm going to crack on with that. I think I'll pull up Erin now. Erin's here. And usual things apply. So remember, guys, please put questions in the question box. Like, we really love answering your questions. It's not even a joke. Like, we absolutely love answering your questions. So just fire away, and we'll come and hopefully, have some interesting discussions that follow from that. Let's resume on Neuroimaging Connectomics. So I'll pull up my screen. And first interesting question is going to be, is my notebook still live? It should be. Yes, it is. Okay. So we did tractography. We computed these things, streamlines, whole brain, white matter pathway reconstructions, all good. So now we're going to go from there to connectivity and connectome. Now, it's actually just one simple step in DiPi, which is this connectivity matrix function here. So I'm just going to run that. And then I'm going to pull these things, plot them in this data frame, and then we'll talk a little bit about what we've got. This runs fairly quickly. So what we're doing here is we're running this connectivity matrix function, which is taking as inputs our streamlines object, and then a few other things, our affine matrix, our APARC labels, and then some options. Now, streamlines object, hopefully we understand now, they're the lists of 3D coordinates that define these streamline things. The affine gives us the space. So it tells us what the, Erin mentioned this a little bit earlier on. So with the nifty images, we have this thing called the header. And the header has a lot of important information. And one of the critical pieces of information in the header is is this thing called a world to voxel, or a world to image transform. So it's a 4-dimensional list. It's a 4x4 array. I mean, why don't we just take a look at it right now? Fine. Yeah, and actually because of the way we did the reconstruction, we actually don't need to do any transformation, so the affine is a bit redundant here. But this 4x4 matrix here is a super important small piece of information in neuroimaging. It tells you like where things are in space in a nutshell, because you have a bunch of numbers in the image, and you have the real world, which has coordinates, and we have this coordinate frame that we typically use where 0 is the middle of the anterior part of the corpus of the anterior commissure. So you need to kind of map this notion of a 3-dimensional coordinate system onto this abstract block of numbers. So that's what your affine is doing. So we need to tell our connectivity matrix something about space, and then we tell it something about the parcellation. We need to convert it to integers here. Don't worry about that. So we run this, and then the output I've just put into a data frame because I like working with data frames. And then this is kind of the endpoint. So actually, don't aesthetically like. I feel like it should be a little bit wider. Should we plot that? So what we get with our connectivity matrix is block of numbers, 2D array of numbers with our brain regions or our parcels. So I made it I plotted it large so that you can actually, so I can see the labels here on the left and the right, so you can kind of see exactly what the rows and columns correspond to. Now, just one or two comments on this on this matrix, because we'll kind of come back to this a little bit. The ordering, number one. So we've got the first half. So you can think about this in quadrants, where we have top left square, bottom right square, bottom left square, and top right square. Okay. The first half of the labels, you can just about see if you look in, are all left hemisphere regions. And then the second half of the labels are right hemisphere regions. Okay. Now this is a choice that we make, just a visualization choice that we make about ordering. We can do different orders. And people sometimes do, like, they pair left and right, in like one region left, one region right, next region left, next region right. But this is an ordering that I kind of prefer, and it's the most common one, where you have everything in the left, then everything in the right, and then within the left and the right, you order them from along basically the y-axis of the brain, which is the front-back axis of the brain. So it's roughly going from within the left hemisphere, going from the front to the back. So we have this, when we have this ordering kind of left hemisphere, then right hemisphere, first half left hemisphere, then second half right hemisphere, that's when we get these 4 blocks. This particular kind of block structure in connectivity matrix, where the top left-hand side of the block, the first quadrant, all of the entries in this matrix here, these are connections that are within the left hemisphere. Left hemisphere nodes, they're streamlines that are connected to other parts of the left hemisphere. Then the bottom right, intra-hemispheric as well, but for the right hemisphere. Okay, and as you can see, a few things about just the structure of this is that the left and right hemisphere blocks, they are a lot denser than the what we call the off-diagonal blocks. So the off-diagonal blocks are the left to right and the right to left. Okay, but because it's symmetric, we just think of it as like cross hemispheric. So symmetry is another thing to talk about. So this is a symmetric matrix where everything above the diagonal is the same as everything below the diagonal. So it's actually all of the numbers are reflected along the diagonal, because we don't really get directional information from diffusion MRI. So when I say the top left block is intrahemispheric left hemisphere, well, it's the top left block, but actually really it's the top left block, everything just above the diagonal line, because everything below the diagonal line is the same as is repeated. It's still useful to think about them as blocks all the same, and if you have an undirected matrix, so if you have a directed matrix, then you won't have you won't have the numbers reflected across the diagonal, and it will make more sense to think about things in terms of like the full blocks rather than just the triangular half of the blocks. So we have more connections intrahemispherically than cross-hemispherically, as in these two quadrants, top left and bottom right, are much denser than these two quadrants, top right and bottom left. What we do have, though, and you can see it a little bit in some of these, there are different, could come out a little bit better, but is we have this thing called a secondary diagonal. So we'll see actually better secondary diagonals, I think, when we look at fMRI data, because they come out really strongly. But we do have some secondary diagonal here. So what you see when you do this kind of ordering is you see this line going down, kind of slicing down diagonally within each of the the second and the so the upper right and the bottom left quadrants. This actually gives you the connections between hemispheric homologs, because it's kind of one region in the left to the same region in the right, right? So you often get this kind of what we call secondary diagonal in the off hemispheric blocks. So we'll come back to these matrices a few times. I want to just comment on the general structure. Another feature of the general structure as well is that you see that it's very bright. The brightest numbers are along the diagonal, and not just the actual diagonal numbers, which, depending on what you're doing, might not mean so much. With tractography, they're not super meaningful, but you can actually have streamlines that leave a parcel and come back to a parcel. So it's not like a Pearson correlation where the diagonal is always 1 and it's kind of trivially meaningless. But still, the diagonal you were kind of less interested in per se. But the key thing is there's this penumbra around the diagonal. So you kind of see there's just a lot more stuff near the diagonal than anywhere else. And what that reflects is the fact that because these are kind of spatially ordered layout, it basically shows you that brain regions that are near to each other tend to be connected to each other. There's this kind of spatial distance function. That's what usually brightness around a diagonal means in these kind of geometrically ordered connectivity matrices. Good. So so we have the full matrix, and we have the tractography streamlines as well. So we can actually pull out some individual connections here. So here I'm just pulling out one. The grouping, as we saw before, this was returned when we did this connectivity matrix function. This is a dictionary, and the keys in the dictionary give us the like row column pairs or the label pairs for each of the connections that are identified. So we're just going to pick one of those, which we just so happen to know is a left-right superior frontal tract. And I'm just showing it here in a kind of slightly artificial visualization way. I just gave it like a very high number to show this is where I'm getting this number from. It's one location, one number in this connectivity matrix. I mean, the value of this number, ignore it, but this is a way of me showing you where it's coming from. So we can take a look at the actual streamline. Again, it's a list of coordinates as we saw previously, and we can plot it as a streamline, as a kind of scatter plot line. And as we saw before, like the name of this thing is left-right superior frontal. Actually, we can see here. So where is this appearing in our connectivity matrix? It's appearing in the off-diagonal block, the upper right quadrant, which means it's a cross-hemispheric connection. Right? If it was in the upper left or the lower right, then it would be an intra-hemispheric connection. But because it's in the upper right or the lower left, again, they're symmetric, then we know it's a cross-hemispheric connection. So I already know that from the fact that it appears here in the matrix, but we've also labeled it left-right superior frontal. If we take a look at the actual streamline, we also get something of a confirmation of that, because we can see, and as I said before, we know what the corpus callosum looks like. This is a callosal tract. It's connecting some part of the left frontal lobe to some part of the right frontal lobe via the corpus callosum. Again, we can plot it with the glass brain and the slice visualizations to kind of confirm. Okay, this is the structure that's corresponding to that single number in the connectivity matrix. What does that number in the connectivity matrix actually mean? It's the number of streamlines in this bundle, right? So if you can see here, right, there's more than one. I mean, actually, how many do we think is in there? I'm actually interested right now. Oh, I want to check. In fact, I'm going to guess it's going to be about 1,000. I just want to know the shape of this. Length of the list. Oh, not so bad. 1,800 or nearly 1,900. So that's how many streamlines are being shown here. And that's what the number in the connectivity matrix is, right? So these numbers, I mean, it's plotting on a log scale, so I logged it because you can see more of the structure, but each of these numbers correspond to the number of streamlines that we found that connected those two parcels, in this case, the left frontal and the right frontal parcel. And I do think it's useful. So a lot of the time with, tractography, connectomics, people, they go straight to the connectome and then they have like no notion of this, the data source. And there's a lot of context, especially things we were talking in the Gather Town earlier on about stroke, right? If you want to know something about if you have some information that's in the three-dimensional brain space, like such as a lesion, for example, or maybe you're doing like the kind of thing we do in my group, we do brain stimulation, or maybe you have some gene expression profiles, or you have some, something that you maybe have in an image, and you want to kind of associate to a connection in the brain, then you have to go through the three-dimensional structure of the fibers, right? So being able to just having the adjacency matrix where you say, oh, A is connected to B, that's insufficient. If I know A is connected to B, and I think maybe there's a lesion here, well, if I don't know the physical route that A takes to B, right, then I don't know if my lesion is going to be touching or impacting on the physical connection, right? So there's a lot. I think there's value in being able to move between your abstracted connectivity representation and your original three-dimensional streamline volume information, particularly when you have some complementary geometrically defined information that you might want to bring into your analysis. Final two quick things, and then we're done on this, is we can also visualize these things using this nice kind of plot connectome function that Nylon has. I'm showing this just as an example because it's common. So something that you see when you work in this field a lot is that people do a lot of three-dimensional plots of networks. And a prominent person, Mikhail Rubinov, said in a talk once, he called these things ridiculograms, as in I can plot this thing in 3D like a network like this. There's nothing you can really do with that. Sometimes there's a structure that you can see in 3 dimensions in this kind of way, like but often, especially when you have like very dense networks with it's just blobs on blobs on blobs. It's just a mess, right? So what I did as a second pass here is actually picked out just the Corpus Callosum entries, right? And then plotted these in 3D. And this is, a little bit more reasonable because it's not so crowded, and I can see to some extent that there's, these are all left-right connections. That's where all these arrows are pointing. So, and sometimes you can kind of color things with modules, and there's things you can do to help, but I'm kind of just offering a cautionary note really on three-dimensional space visualizations. I mean, I'll distinguish this from the tractography, right? The tractography streamlines, these are the actual the fiber pathways that are we're reconstructing. These things are lines we're drawing in 3D space, right? This line doesn't exist. This isn't a real thing. This line is representing the actual fiber tract that is maybe going via the corpus callosum there, and it's convenient to draw a straight line when you want to do a three-dimensional representation of a network or a graph. But again, depends on what you're doing whether or not it's useful to do a three-dimensional plot. They certainly look good when you do them a little bit with slightly fancier visualizations than this. Can be used for, I would say, about more than half of the time they're kind of visual window dressing, which pros and cons to using that. Okay, guys, that's it for this session. So I'm going to hand it back to Erin to pick up on Fmri.
I'm back. All right. Thanks for that. I'm going to start actually. Wait, I'm going to go back to the slides. going to start the Fmri section actually with first I'll introduce Fmri, and then I'll recap what we did in DWI, and we'll talk about how it's a little bit different, which is maybe a nice lead in. Okay. Actually, maybe I'll go straight to this one. So this morning or in the last 15 minutes, we talked a little bit about this idea where you start with MRI. We start with MRI, some idea of where brain areas are in combination with the DWI scan. You magically, via some preprocessing and math, get this tractogram or this hairball that has all of this mathematical model of where all the connections are, and then you apply this parcellation. And from that parcellation, you count how many of these hairs or streamlines connect to brain areas, and you map that as a huge matrix. Now, going back, we're going to talk about Fmri now. Okay. Talk about Fmri. So Fmri or functional, I don't know, we call it functional MRI or magnetic resonance imaging. Sometimes we refer to it as bold imaging, which is maybe a bit more realistic. So bold stands for blood oxygen level dependent signal. And it's really important to understand that when we're talking about MRI, this is all we have. This idea that came in the late 90s, someone realized that deoxygenated blood and oxygenated blood actually have slightly different magnetic properties. So if we actually capture a movie of MRI signal that's focused on finding this difference between oxygenated and deoxygenated blood, we start to see changes over time. And this appears to be because actual blood flow in the brain seems to change to some extent with the activity that's happening in the brain. So one thing that we realized, not we, that science in general realized is actually some parts of the brain, when you tell them to do some work, sometimes when you tell them to do some work, when you give them a task, like you give somebody something to look at, you see the back of the brain, which has the most important part of the brain for actually being able to see, the occipital cortex, gets increased blood flow to that region. And we can actually measure that. So we can do tasks and we can see increased blood flow. And we can be like, oh, look, the back of the brain is important for looking at things. And that started this huge field of putting people in scanners and making them do things and seeing which part of the brain had increased blood oxygenation to it. And that's this idea of task-related fMRI. And then around the knots, we realized that tasks were too hard to make clinical populations work. And we couldn't figure out which tasks to get clinical populations to do en masse, or to get large studies to do en masse to study. So we decided no task was a great option. So we put them in there. So we put people in scanners and had them just lie down. And we called that the resting state, because they are resting or just sitting in a scanner very boredly, looking, either closing their eyes or looking at a fixation cross. And the reason why this turned out to be really interesting was that in the late knots, I think it was around the late or early knots, some people started to notice that this bold activity, so this fluctuation in blood oxygenation level, was correlated across brain areas. So one first example is your left motor cortex and your right motor cortex. If you look at the time series of those fluctuations in blood oxygen level, you see, if you look at the time series of those fluctuations of blood oxygen level, you can do a very simple correlation, like a Pearson correlation, and you get the other hemisphere, the left motor cortex. So this gave us this idea that if we just correlate or look at how these areas in the brain are correlated with each other, we can build these other types of connectivity matrices of essentially just covariance in this bold signal in the brain. So what we do in fMRI or in resting state fMRI is we take some functional MRI, so this movie of blood oxygen changes in the brain, and for every point in the brain, so every voxel, we get a time series, which is like squiggly lines across time. If we correlate these two things or correlate these things across brain regions, in this case, cut up the brain, average the squiggly lines across axons, and then just correlate all of those squiggly lines, we can get matrices. And those matrices have interesting properties, where parts of the brain that are actually quite far apart, say this one and this one, might be highly correlated with each other. And we might call those two parts of the brain parts of the same brain network. This really took off in psychiatry, this idea that now we have this huge matrix of information that we can get from a person, and can we use this to, say, diagnose or predict treatment response? So there was this whole field in, wanted a precursor here by saying that this arrow, which in this picture is an arrow, is hours and hours of preprocessing, usually. And I didn't want to go too much into preprocessing here, except to say that hopefully in the modern day, unless you're working at an imaging software development lab, you're using a BIDS app container to preprocess your data, because data has now moved into a magical place. We're in a magical, the future is now, where all you have to do is name your files properly, and a Docker container will parse what data you have and figure out the best preprocessing pipeline for it and run that preprocessing pipeline. And it all works, and it's amazing. Okay, so back into, can we use Connectomics to predict treatment? So this is actually a very recent article at the link provided here, which is a meta-analysis of studies that have tried to use these connectivity matrices, for the most part, these connectivity matrices to predict treatment in major depressive disorder. And they say they have decent prediction, and they actually get higher prediction for higher prediction with resting state connectivity for treatment with electroconvulsive therapy than they do with treatment with drugs, which there might be reasons for that. But one caveat is in this whole field is it's very new. So people are actually, all of these studies trained machine learning models using only scans from potentially only 16 to 20 participants. And if you learn anything about artificial intelligence, the first thing you should learn is do not try to do it when you only have 16 to 20 participants. And also, the second thing you might learn if you learn about artificial intelligence next week is that leave one out cross-validation, which is what they did here, which is the weakest form of machine learning validation. So there is to say that there's been people who've tried to do this, but the data hasn't really been there yet. The strongest paper that tried to use Connectomics or use these connectivity matrices to predict treatment response actually came out by Drysdale in 2017. It was very exciting. There were actually some UHN or Toronto Western collaborators on this, and they claimed that they could get they could actually use the connectivity matrices to subtype participants, and some of these subtypes may have been better responsive to DMS therapy. This was an exciting paper at the time, definitely made the author's career, but there's been some controversy about it as it went forward to say that its methods weren't strong enough. And then another paper that used actually slightly better methods now was published in a much lower ranked journal in the recent bit. So there's still not that much data to train these types of models, but there's some hope that models based on resting state might be useful in predicting treatment response. And models based, I should say, this is specifically in major depressive disorder. There's also some work trying to do this type of work in schizophrenia, and I think in the schizophrenia work there is much stronger signal, and there's potentially something there to look at. Okay. So the other area where bold MRI has started to really impact clinical science and potentially impacted a lot more has been in this idea of targeting of TMS, or transcranial magnetic stimulation, or different stimulation therapies. So actually, I should ask John if you want to comment on this, because it's also something you like.
Yeah, I certainly can. This is super exciting work. Oh, Erin dropped off. Maybe she'll come back because the figure is kind of handy. So this group, Mike Fox's group, have been doing this. Erin, you're back. And Erin, do you want to pull up the figure again? So they've been doing this work with TMS and depression. They do actually do a lot of things, but one of the things they've been doing is this work with TMS and depression, where they've been trying to figure out which places where they target in this part of the brain, the dorsolateral prefrontal cortex, where most people go to try and try and do depression therapy, TMS treatments. They found that it seems that the parts of the DLPFC that have the best efficacy in terms of treatment outcomes are the ones that are anti-correlated with the subgenual cingulate in fMRI functional connectivity maps. So exactly. Well, actually, Erin, if you just go back, stay on the previous slide for a second. So the background map here, the kind of blue and red, that's a seed-based functional connectivity over the surface vertices with a subgenual cingulate cortex seed. So when the color is blue, it's negative, and when the color is red, it's positive. And the point is that the points, the kind of dots that they're overlaying there, the ones that fell in the negative correlation part of that map were the ones that had the best success rate in terms of the depression remission outcomes. And that notion of a functional connectivity and negative functional connectivity, I think that's when maybe, Erin, you might want to go on to the next slide. Maybe you want to pick up and elaborate on getting at there.
What this is?
Yeah.
Yeah. So so subgenual cingulate cortex, if you're is the yellow dot here, and so they took bold activity from the yellow dot here. If you look at it in the human, in people, you get a time series. So this red line. If you look at functional connectivity from this green circle here, you get this line here. And this is an example of it potentially being anti-correlated, because you see a subgenual cingulate is going down, DLPFC is going up. What they showed in this is they were looking across multiple studies that had tried TMS targeting in the past. So every dot here isn't a person, it's a study. So it's the targeting location that was used in general across a study based on the targeting technique they tried. And if they compare across studies, it seems like the ones that targeted an area that was most anti-correlated with the subgenual cingulate cortex, they tended to see better treatment response in the study as a whole. That's this general finding. So there's a lot of current work going into seeing if we can actually do this in individual participants. So if we can build pipelines to try to target the subgenual cingulate cortex in specific people, or to try to target the part of the brain which will give us the best depression treatment response in specific people, can we actually improve treatment response to TMS? That's a pretty cool clinical application. And I don't know if we want to pause and talk. Oh, wait, but before we go into the notebooks, where I'm going to show you how to calculate things, I want to give a shout out to pre-processing techniques, and also a shout out to Jerry, who's been speaking, and because this is totally his slide. So why we pre-process fMRI? There's a couple things that need to happen before we can start analysis. First thing is that fMRI images give us a huge cube or a shoebox fit around your head, but we just want data from brain areas. Also, the problem is that the fMRI and the T1 weighted anatomy sometimes isn't aligned to each other, and doing that can be very difficult because fMRI is more fuzzy. fMRI, this is a large problem, gets distorted, especially around the area of your nose and your ears, because that's where air pockets exist in your brain. And sometimes we have some techniques that can help with that distortion a little bit. Participants and scanners move, so they're not actually even aligned to themselves. And movement actually influences the fMRI signal in bad ways that we sometimes need to account for additionally. And participants aren't aligned to each other. So So everybody puts their head a little bit differently in the scanner, or the scanner aligns their head. Doesn't actually do very little to align their head. So we need to align the head to a template so that we can actually compare across people. So there's some important steps here. Here's an example of a raw fMRI image. Importantly, we need to find the brain. Surprisingly, the hardest part. We need to correct for motion, so people moving in the scanner. We need to correct for susceptibility distortions. Actually, this is a cool GIF because you can see the distorted areas kind of moving in and out, especially around the cortex in the front. We need to align the fMRI image to the anatomical image. And we need to transform the data, and we need to do this idea of confound regression to try to get rid of noise signals that are caused by motion. And all of this is hard. And in your entire PhD, you will probably learn about some of these things, but you might not actually learn about how to do all of them. So a magical thing that happened in the last 5 years is that people built BIDS apps or pipelines that combine all of the best practices or best-ish practices for all of these steps and put them in Docker containers. And now everybody just runs the Docker container. And the Docker container is smart, so it doesn't just read the data. You don't just give it a file and you go. You actually give it your entire data set. And your entire data set is labeled in a way that it can figure out what are the anatomical images, what are the bold images, which ones were done together, which ones were done apart, and try to preprocess them all in the best way. Also, there's a discussion in the chat about naturalistic stimuli, which I totally agree with. Watching movies is probably better than staying thing. But I also think it's funny that we consider naturalistic stimuli as just a lot of screen time, like scanned screen time. So nature for us is looking at a screen or looking at a video. Okay. I got to give a shout out to the data that we've been using in the structural tutorial and in the fMRI tutorial today. So it's from the Midnight Scan Club, which is actually these guys plus 4 more, 10 people. But literally, they were MRI scientists who decided to stay up till midnight when the scan time was super cheap and scan each other a bunch of times. And they do a bunch of sophisticated analyses on the scans that they got. And what they got was careers out of a very cheap amount of scan time. Enough scan time, you don't even need a grant for that. But yeah, they got 16 publications, including 5 Neuron papers and 3 PNAS papers, which, if you know the neuroimaging literature, that's actually that's career-making publications there. A ton of citations, because they did a lot of research into what's the same amongst these 10 people and what's different, which is kind of something that we hadn't really thought about. Also, they put the data and the preprocessed data on OpenNeuro, which is this amazing resource, which actually I should click on at this point. Because PS, if you want to get started in neuroimaging research today, go to OpenNeuro. So OpenNeuro is a resource of both actually MRI, but now also MEG, EEG, ECOG, everything data. It has a public dashboard, and all the studies that are in this public dashboard, which I think are now thousands of studies, have data that you can download today and get started with. And it's all organized in a very similar way so that you can understand it and you can get going right away. So what we're going to use is the Midnight Scan Club data, so the data from that experiment where people scanned each other. And the nice thing about this one is that in the Midnight Scan Club, which is the bottom one here because it's the locust. Wait for it. Oh no. Okay, in this Midnight Scan Club data set, you can browse data right away. you can learn about it. You can watch data sets, so you can like receive notifications if anything changes with the data or if they find anything that's wrong. You can also, they made available this idea of this folder called derivatives, which is what we're going to work from. So they already pre-processed the data for us and made it available for us to download. So that's what we're going to work with today. And okay. So with that said, I'm going to go to our, if I can find it, I'm going to open and collab our Fmri notebook. And this one is notebook number 3 of Neuroimaging Connectomics. Fmri connectivity, this one here. And remember, we're in Google Colab, so actually the reason why this only works in Google Colab and the Docker is because I added this weird extra bit to install something called Connectome Workbench, which is really great for surface-based analyses, to do some steps which don't work as quickly or nicely in pure Python right now. I'll go over what those steps are. Okay, so the data, so we're pulling some data from the Midnight Scan Club, which is why the subjects are called MSC here. I'll just let that go and start talking about what this is, and then let's see. Yes. And then the extra little thing that I downloaded for you guys was this one little executable called WB command, or workbench command, which is this executable published by the Human Connectome Project team to work with this new file format that the Human Connectome Project has been pushing, called SIFTY files. And actually, a lot of SIFTY file computations we can do in pure Python now, but there's one step that I actually really want to show you because it creates this new file type that I really like. And we'll go into that a little bit in a second. So for today, we're going to be using the classic science libraries, but we're also going to be using NIBabble as nib, and we're going to be using the NiLearn plotting functions as well. Okay. So the reason that that I made things complicated for myself and made this this tutorial use Connectome Workbench was essentially all for this step. So in this step, we're taking those two label Gifty files and we're actually combining them into one file called the SIFTY file. And this is just useful because it combines left and right label data into one file that contains both. And we're using that file to do something called SIFTY parcellate, which means we can take the functional data, which has already been mapped to the surface, and we can parcellate it or cut it up with the atlases that exist in that APARC or Free Surfer Atlas that I just showed you in the beginning earlier today. So when we do this, we get a file that ends in pt-series.nii for parcellated time series. And it's weird. It's a new file format. Not as many people know about it. I could have given you a simpler version based on Nifty, but I wanted to show you the PT series file because it actually makes some of the other computations that I show you in this notebook actually a lot easier and a lot more straightforward. So if we load this PT series file, we can load it with NIBabble. Once we load it, we can get the data like before as a float, and we can look at its shape. We can see that the data is now just a two-dimensional array. So the nice thing about PT series is now you don't have to think in 3 dimensions because all we have is, you can think of the data in PT series almost like a spreadsheet. And actually, usually time series data, I used to just store it in a spreadsheet in CSV files. But the PT series file format has two two big useful pluses on top of just storing time series in a spreadsheet. One is that we can get the spreadsheet back by loading in Nibabble, but this file, the PT series file, is actually a lot smaller on disk than it would be if the same data is written to a CSV, just because the data is compressed. The other reason why I like the PT series file is that the PT series file actually contains not just the data, but the information about where the regions of interest or parcels exist in actual space, and what the name of those parcels are. And we can get those out of Nibabble using these commands. So we've loaded this PT series file, and we notice it has 2 dimensions. The first dimension, this 818, is time points. So that's how many time points, how many frames there are in this video of the brain. This 34 here is for the number of parcels or regions of interest that exist in our time series. If we want to get the names of those parcels, it actually exists in the header of the file. So we can pull it here, and we can see the names of those 34 brain regions. And we can put that data together really quickly into a Pandas data frame. And just think about it as a Pandas data frame, because it is 2-dimensional data. So in this case, I've written the name of the brain region as the column name in the Pandas brain region in the Pandas data frame, and every row in this Pandas data frame is a time point. The nice thing about that being a Pandas data frame is that now we can do computation on it really quickly. But I also wanted to backtrack and tell you that this PT series file, or the reason why I like it, is that the PT series file also contains information about where in the brain this information came from. Oh wait, so one thing we can do this is just taking 2 columns from that Pandas time series, from that data frame, and plotting them. So if we plot them, we can see those time series, and we can label them with the column name of the data frame to be able to see. So do we think these are correlated or anti-correlated? I guess we can see as we go. It's kind of hard to tell here. But we can also look at the location of the file. So we can run this. So the information about where exactly the regions of interest are things that we can actually pull from the header. So we can use this little bit of code to kind of pull and plot the actual location of a region of interest based on the header of the PT series file. So we can see that this area called rostral anterior cingulate, which is the time course that I'm plotting in orange above, actually came from this part of the brain, which is pretty cool. Okay. And that's nice because that's being passed around in a very small file. Okay, the nice thing about we use this information to map it to a data frame is that there's a one-liner computation we can do on data frames called .core to actually create a correlation matrix out of the data frame. So we can run that. It's like 2 seconds, and we get a correlation matrix, which is region of interest by region of interest. We see that regions of interest are infinitely correlated or perfectly correlated with themselves. But some regions are correlated with each other-ish. So we can see there's a correlation minus 0.44 between the insula and the caudal middle frontal cortex. We can take this data frame and we can actually just feed it to Seaborn, which is just a plotting functions that I like because they work the same way as R plotting functions, and they make my life easy for data frames. And we can plot it this way. So this is literally just all of these numbers are now represented by a color when we look at the matrix. And then the nice thing, because we had the p-scalar file, humor me, I wrote these 2 ridiculously large functions, but they do 2 things that are kind of easy, or not easy. They do 2 things that are kind of very useful when we get to the plot that I'm going to get to below. The first thing is that we can take any column. We can use this first function to take any column. So in this case, we're taking columns from this data frame, and we can write them back into that p-scalar format, which is kind of nice. We can attach the information about where the parcels were to our result, so that we can save it for later, or so that we can plot it right now. So, and the way we do that is by literally copying the information about where the parcels were from the original input file. So that's long, but hopefully it makes sense. The second function that's here is kind of nice. It takes that parcel information and it tries to map it to the cortex. So it takes data and the parcel information and it creates a long vector to map all of the individual result information to individual vertices on the surface. Cool. Okay, so those were two long functions, but I just want to show the two in combination here. So we can take our result and our original input file. In this case, we're taking not the whole result, but one column of this data frame. So that column with the column name roster anterior cingulate, and we're going to map it to the surface and plot that. So wait for it. Nice. So what we're plotting here is literally one column, or literally only the values in one column, the column called rostral anterior cingulate. So all the values here, and we're plotting them back to their original surface locations. Right here. So we can use this to try to figure out across the brain what is correlated with this ROI, the rostral anterior cingulate. And we can actually see a nice area of anti-correlation in the dorsolateral prefrontal cortex. So this might be where we want to put some TMS stimulation. But this is actually, those are some pretty big ROIs. So maybe this is not the best model of exactly how we want to start calculating connectivity. So instead, I'm going to pull a different parcellation, in this case, the Schaefer parcellation, which has smaller parcels. I'm actually going to pull two different versions, one where there's 400 parcels and one where there's 1,000 parcels. And I'm going to start with the 400. I'm going to run the sifty parcellate command to parcellate the file. And then when I load it, I see that now I still have 888 points, but it's by 400 regions instead of 34. Then I'm going to redo, and notice that I still have the names of these 400 regions. This is the first 30, and they have a lot of weird, they have weird names, but it's because they're based on functional connectivity or this attribution of networks to these regions in the brain. And they don't necessarily map directly to anatomical connectivity. At some point, they just start labeling with numbers and giving them random names. So then we can get back our heat map. And now we can see a lot of dense, interesting information. Also, we can go back to remember what John was saying about the structure of the structure of connectivity matrices we see in functional data. So in this case, we have all of the left regions and their correlations with themselves, all of the right regions and their correlations with themselves. And then this red line we see here, this is the off diagonal. This is connectivity across contralateral left-right regions of the cortex. And we can see in functional MRI how some of these contralateral areas or left-right correlations are actually some of the strongest we get in Fmri matrices. And then we can do the same thing to plot that back to the surface. And here now we're using this ROI to calculate correlations with the frontal cortex. And now we see actually not very strong functional connectivity, except maybe some decent anti-correlation up here in the top in this particular person. And so now that was with 400 parcels Schaefer version. I'm going to move to 1,000, and I'm going to say 1,000 because so we're going to build 1,000 by 1,000 connectivity matrix. We can go in this data set as high as like 32,000 by 32,000, but we would crash our computational engine, and we would get something that we couldn't really might have gone a little bit too far in terms of actually trying to figure out what's going on. So this is when we cut the cortex or the brain or the cortex up into 1,000 bits. If we look at what's correlated with this part, we actually see kind of tiny areas. Maybe we would want to put our TMS stimulation here now because this is the most anti-correlated region. And this is terrible because I'm not very good at margins, but you can see how actually with 400 and 1,000 we get kind of very similar ideas of network structure here, but we get something more fine-grained with 1,000 and 400. Oh, and one kind of bonus area is that I showed you how to do correlation. I'm showing in this notebook how to do correlations all completely in Pandas, but there's this entire library called NiLearn, which is magical and does all of these different other types of connectivity and all of these different types of machine learning models and plotting. And it has some beautiful tutorials about calculating different types of things beyond Pearson correlation. So you can do also partial correlation and tangential. And this is just a little bit of how you would do the exact same thing, but using NiLearn's fit tools. And I should also say NiLearn's fit tools are also really nicely computationally good. They're built to run on small systems, so they do a lot of really nice in the back Python tricks to try to make it work on everybody's computer. And that's what I was going to say about functional connectivity. So I'm going to stop sharing. Let's see if we have any questions. So one question is, oh, is it from Amin? I don't know if John has already taken this one. It says, regarding correlation matrix, what were the values? Bold signal. So if positive core between two regions, this means that bold signal increase in time series in one region is correlated with an increase in time in another, or did I misunderstand? Yes, that is exactly what it means. So yeah, Min, you got it. In this case, all these examples, we literally just did Pearson correlation. So when it goes up, the other brain region also goes up. And when it goes down, the other brain region also goes down. We're literally just taking those two time series and then running a Pearson correlation on them.
I guess the one thing that we should add that needs to be at least mentioned when we talk about positive and negative correlations with fMRI is there is a it's actually similar. There was a discussion on the thread about movement, right? So there's a few in all areas of science, but in neuroimaging, there's a few kind of technical areas where there's like there's dragons, let's say, like there's challenges that we know can kind of be misleading, but so negative correlations is one of those areas as well. It actually because it's to do with this thing called global signal regression, which is when you take out, you subtract the mean from your fMRI signal, which is kind of in general a perfectly standard thing to do with a lot of statistical data analysis. But when you do that, you actually introduce a lot of artifactual negative correlations, and it's been kind of demonstrated robustly that is the case. However, there also do seem to be genuine negative correlations. So it's this murky territory where we want to see these things. They're probably there, but we also are aware that a lot of the kind of standard processing approaches can potentially make us think that we're seeing things that are false positives in terms of looking for negative correlations. So global signal regression negative correlations is, you know, a long conversation, but we should at least kind of give it its dues.
Yeah, I guess the least controversial way is that it seems like blood flow, I don't know if they're negatively correlated or if they're just not correlated, and the whole mean has been shifted down. But in that way, they're considered almost functionally the most separate of brain areas. So either they're just not turning on at the same time as each other, or they're turning on entirely independently. But it's kind of weird that the thing that's the at least least associated with subgenual cingulate is the area that seems to be a decent TMS target.
Yeah, I mean, it says why skeptical of negative correlation specifically as well. I mean, so it's actually kind of extending past the neuroimaging connectomics, and actually from the whole brain modeling, my whole brain modeling hat. Negative correlations are an interesting thing to see, and from my point of view, it's interesting to see negative correlations at slow time scales in neural activity, because when you have systems that have, you know, fast dynamics, then it's not surprising to see negative correlations, because it's kind of networks that interact with each other, they tend to produce kind of anti-correlated modular blocks. It's a, you know, perfectly standard thing. But the negative correlations in fMRI are on a really slow time scale, and in general on time scales that are slower than the, what would you call it, the characteristic time scale of a system, those kind of negative correlations are a bit more unusual than things tend to kind of globally correlate. That's what you kind of would expect actually on slower timescales, at least my understanding of how physiology processes relate to like slow timescale fluctuations. So we're still trying to get our head around how to understand negative correlations, but they probably are there, and they probably do mean things.
Cool. And actually, that's a nice segue into the next section where we're going to talk about EEG, and we're going to switch timescales and we're going to look at electrical activity as opposed to blood flow in the brain. Because one thing that's really important to realize with fMRI is that these videos are actually really slow. Each frame is between 800 milliseconds and 2 seconds. So when we're talking about a correlation, we're talking about a correlation in the ups and downs of blood flow over the course of like 5 to 10 to 15 minutes, versus EEG, which is much more condensed, much closer to the timescales that the brain actually works in.
We had another question, so from Faranak, which is, how can we, well, we had the segue, Aaron, but maybe we'll take the question anyway, how can we extract the exact coordinates of, sorry, I'll do this, I'll cue start answering. How can we extract the exact coordinates of the regions and match them with the white matter bundles from DTI, e.g., should we use one atlas to parcellate both DTI and fMRI? So I think I can take this, but Erin, you can add any comments. Like, yes, we use the same atlas, but we use it slightly differently, in that if you think back to the surface plots that Erin was showing before, so the surface-based parcellations in particular, there's so if you think about that ribbon, the cortical surface, the top part is the pale surface, the bottom part is what we call the white matter surface. When in terms of fMRI, the thing you're interested in is the bit above the gray white matter interface, where the kind of gray matter ribbon is. With diffusion, you're more interested in the bit just below. So if you imagine a little kind of part of a little bit of a cortical surface there. Like with fMRI, you want to take, I'm really looking for something around to kind of represent this. Yeah, this will do. So if I have a little bit of cortical surface there, okay, with fMRI, I want to be taking signal from there. Whereas with diffusion, I want to be taking signal from there. If you can imagine, this is not filled in. right? So the point is we use the same parcellation, but we kind of nudge it we nudge it more towards the gray matter when we're doing fMRI, and we nudge it more towards the white matter when we're doing tractography. But in general, we aim to use the same kind of parcellations because then we can think about networks in a complementary way.
But there's a caveat there too, because these different modalities have different scales that they work better at. So different sizes of brain regions that they work better at. So DWI works nice with the APARC automatic parcellation and tends to get used a lot with automatic parcellation. But if you see an fMRI paper that focuses on the automatic parcellation, I don't like it.
Right.
For the reasons that I plotted in the file, you can see that two different brain networks usually exist within the same parcellation of APARC, so it doesn't work as nice. And it's a huge caveat when you try to do multi-scale to figure out how to combine these things. Yeah,
at this point in time, it's only like in terms of the state of the art, or not really the full state of the art, because that's kind of 7T, but kind of the trend in terms of what most good data sets are. The resolution you can push parcellations and connectomes for fMRI is higher than you can push parcellations and connectomes for diffusion. It's like you can hit about, well, at least with the originals, it's a function of the spatial resolution in general, but so things like the 32,000, the dense connectome, the 32K parcellations that Aaron mentioned but didn't really show, they kind of work with fMRI. I mean, they're huge. They're really difficult to work with because they're like 32 gigabyte files, and it's really hard to do stuff with them. But you can look at correlations between things at a very small scale with fMRI, and it's kind of okay. You can't really do that with diffusion. You can't go that low because it's just going to be an extremely sparse matrix. Like you need to kind of average over a certain amount of, or you need to kind of give a certain amount of space where those streamlines can come in, and the streamlines, there's only so many streamlines. They don't go everywhere. Whereas with fMRI, you have a time series at every single location, so you can. There's no question of like you having a time series for a given small resolution unit. But with diffusion, you're not going to have streamlines going into every single voxel. They just don't have that spatial resolution typically. So 1,000 nodes is kind of a good upper limit for diffusion tractography. It kind of starts to get a bit iffy about there. People generally go with like 200 to 500, and certainly not 32,000. That's challenging. At the end of the day, you'll just have a very, very sparse matrix, even if you do probabilistic tractography. Good. Okay, so we also had a queue. Okay, so while I'm in the questions box, before we move on to the final tutorial session, we had a question that I'll just kind of comment on this question. It was well answered by Jerry, but I actually had one or two additions. So the question was from Mohammed, and it was, since we are measuring water diffusion, why don't we get directional information, as in which region is sending? So this is in terms of tractography, as in which region is sending and which region is receiving axons, and whether they're running in one or both directions. And the answer to this, so the first part of the answer to this is Jerry is actually given quite well in the comment, but just to kind of summarize the orientational information that we get with the diffusion, what we call the tissue model for diffusion, those ellipsoids that I showed you at each voxel, that's symmetric. So, and you should think about it as kind of each of those little ellipsoids, there's kind of a drop of, you could think about it as a drop of water in a little pool that diffuses symmetrically and gives you some idea of the shape of the space, but doesn't kind of, it's symmetric. We'll just kind of leave it at that. It's kind of diffusion in all directions, right? So that's that one part of the question. The answer to that question is exactly that, which Jerry said. But I'll add the other part, which is the kind of macro extension of that, because that point about the symmetry of water diffusion, that applies at the voxel level, okay? But the question is actually directionality at the fiber tract level. And this is really more to do with tractography. So it's kind of what we're doing is we don't have what we have is this direction field, right? So we can move what we're doing with the streamlines is we choose a starting point and we move it through the direction field, but the direction field doesn't tell us which should be the start point and which should be the end point. Like, it should give you the same thing going this way to going this way. If it doesn't give you the same thing going this way to going this way, it doesn't mean that there's an asymmetry in the connectivity. It means that there's something wrong with your algorithm, because there isn't anything, there isn't any information about start and end points. If you think about an axon, an axon, sure, it has a start point, cell body, and then it goes to where it goes. But that we don't have that kind of cell body to projection level information. We have more like a piece of string that you can kind of choose to go that way to that way, or that way to that way.
Yeah. I guess one thing is to remember in diffusion, we're trying to map the white matter. We're trying to, especially like deep white matter, we're not mapping the gray matter at all. So we're not actually even capturing the end point and the beginning point. We're not capturing those cell bodies. We can't find them.
Exactly.
But I guess one pointer, there are ideas like the COCOMAC database that tries to kind of create general connectomes based on actual macaque or preclinical non-human primate data, where real tractography experiments have happened and they've actually tracked the entire neuron.
Yeah, exactly. So the gold standard for kind of methods comparison essentially for these kind of tractography data is indeed what we call like invasive chemical, typically ex vivo track tracing. So it's normally kind of, you have a macaque or sometimes they do in other non-human primate species, and they sacrifice the animal, and then they inject these chemical, actually no, they do the injection before they sacrifice the animal, because they need it to diffuse for a certain amount of time. But it's a destructive operation. But that kind of it's very hard work, right? So you have to do lots of experiments. But that research happens, and we have that information in limited context, and that's the gold standard, because that is directional. You inject this kind of chemical, this dye, into a certain location, and it's uptaken by the cells, transported along intracellularly along the axons, and then deposited at the axonal cleft. So you actually have a directional information there, and that is the gold standard. That information, those data sets exist. They're rare and expensive, but they're extremely valuable. The best data, the gold standard, is actually at this point in time is coming from a group in France. By the lead's name is Henry Kennedy. So if you're interested in like the best available non-human primate connectivity data, go Google like Kennedy connectivity, and that'll probably take you to some good resources. So we try and benchmark the diffusion data against things like those data sets, and also things like data that's kind of acquired in humans, but where, in cadavers, where they basically take a cadaver, put a head in the scanner, and then they do diffusion, but they do a diffusion MRI scan for like 60 hours and get lots and lots of very high quality data. So that's kind of in between the chemical data and the kind of things we can do in vivo in humans. It's kind of cadaver diffusion MRI, which is also very, very good data, and it's very interesting what they can find with those kind of samples. Okay, good stuff. So we will move on to our final tutorial of our morning session, which is EEG, MEG connectivity. So I will share my screen and we can crack on. So I don't actually have a lot of extra an extensive amount of slides to go through in terms of rationale, and I think given the time and so on, we'll just move straight to the tutorial. So just going from the repo again, and I'll just kind of go through the process. So open in Colab. And then we're at day 5, and it's going to be 04, MEG connectivity. Okay, now the reason, so this is this modality completes most of the picture of not only connectomics but also neuroimaging in general. And it's kind of, I'm going to kind of use the right wording here. Firstly, depending on who you talk to, EEG and MEG is not neuroimaging, and from a certain point of view, that's strictly speaking true, in that you don't, the kind of basic data aren't images in the same way that they are with MRI and Fmri and so on. But from a practical standpoint, yeah, for at least from speaking personally, like I count EEG and MEG and other related things like FNIRs within the cluster of principally human non-invasive techniques, but also available in other species as well. Include EEG and MEG in there. And if you say combine, say Fmri, EEG, and MEG, I think that's going to be well over 95% of all of the current and previous research in this area. So that's what I mean when I say completes the picture. It isn't complete. I mean, there's PET, there's FNIRs, there's ECOG, there's a bunch of other things you can do with MRI, like ASL. There's interesting things with depth electrodes and so on. So there's lots of things that is not included in just Fmri, MEG, and EEG. But as I said, it kind of covers most things. So it's a good place to wrap up. What we're going to do here is we're going to, again, go through a tutorial. I'm going to give some comments. It's going to be, I'm going to make some nods to the methodologies and some of the elements that we've looked at previously, the connectivity matrices, parcellations, etc. The tutorial that I put together here, again, is based on another tutorial, and I'm not claiming authorship of any of these things in a kind of fundamental sense, and why would I bother doing that? There's good resources. So the tool that we're going to be using here in the tutorial this is based on is a software library called MNE, and then we'll just quickly pop to there's the link. The correct link is there in the docs. It's from the development version, bear that in mind. So you won't see it if you're in the non-dev tab here. But I want to just mention, give a nod to MNE. So this is another neuroimaging analysis tool, and this is there's a lot of players in the space. There are a lot of software libraries that have been around. If we're talking about EEG and MEG specifically, the old school like OG tool is EEG Lab, and then other ones like that are commercial, like Beezer and Curry, and the other big player in big widely used software library is Field Trip, which is the MATLAB toolbox. So, and also SPM is a neuroimaging library that has a lot of good functionality. Other ones like Brainstorm. Sorry, I know I'm just listing off names here, but I want to give kind of dues to the general landscape. To my estimation, according to the things that I find useful and so on, but also in terms of the community component, what I think is an important part of open science and open source software, that MNE is the best tool for doing EEG and MEG analysis. I can expand on the reasons, but I'll just kind of leave it at that for now. I strongly encourage people who are interested in learning more about this part of neuroimaging and neuroimaging connectomics to Same with DiPi actually, is just hit this website and just go through all the tutorials, and you learn so much from these tools just going through. They have such a rich, there's like, I think probably about 40 tutorials now, like examples, code-based examples. You really get a good exposure to all parts of the process by spending like a few days working through the tutorials. So if you want to go further than what we talk about here, then this is the place to go. Back to our tutorial. So we're in Colab again. Oh, this is the right one. Let's do this one. Yeah, this is right. So as before, we need to do this pip install, and the only non-standard library here we're using is MNE. Actually, Colab recognizes that I've already been here in my browser, but I'm starting a new instance. So we're going to install MNE. The rest of the stuff we're going to use is standard it's called scientific Python stack. So we're just going to import, as previously, some visualization things and some MNE tools. This is a label reordering that I happen to prefer, that reorders the connectivity matrix. We'll see that in a little bit, but it's just a variable that we need to define. This line here, so this is pulling the data. So similarly to, or the first part of this is pulling the data. So similarly to the DiPi example, if you're running this for the first time, it'll grab some files and pull them to your location, and that'll take a bit of time for the MNE one if you're running in Colab, because it's about 1 gigabyte of data, but it'll finish in, I think, under a minute. So hang in there if you have a progress bar that you're waiting to go through. So we pull in our data. Now, what are we looking at? So this data set is actually a task. It's an audio-visual task. And we're actually not going to be thinking about the audio-visual task part of this, which you would do in other contexts, but we're actually just going to treat it as something we can pull time series from. Okay. Now, with EEG and MEG, our starting point is channel data. Okay. Starting point is channel data. Extracranial channels on the scalp for EEG, or kind of slightly above the head for MEG. And what we want to do, not all the time, but particularly for the things we want to do, is we want to estimate source what we call neural activity in what we call source space. We want to estimate the locations of the activity in the brain that is giving rise to these fields that we're measuring, a couple of centimeters away from their original sources. And Frank already described a fair bit, a little bit of the framing for this is at the level of a single dipole and a source far away. So actually there was some reference to source analyses as well. So this is exactly what we're going to do. So we're loading some data. We have some event info, and the actual data is the raw data, and we have this thing called an inverse operator that I'll talk about in a second. Now, what is this raw thing to start with? Well, it's an MNE object. It kind of has information in it that will tell us about the sampling frequency, and in this case, it has EEG and MEG channels. This is a common thing to do if you have you can record EEG concurrently with MEG, which is quite a powerful thing to do because you have two modalities and like a lot of extra data. So we have some EEG channels. We also have these two type of MEG channels called magnetometers and gradiometers. Gradiometers are they are more sensitive to sources that are closer to the sensor, whereas magnetometers are more sensitive to deep sources, to do with the orientation of the field that they measure from, whether it's radial or perpendicular. And so we have our EEG channels, and then what we can do straight up with just with some native MNE functionality is take a quick look at the data. So if you're running this, if you're running this like locally, you can actually produce an interactive visualization. You can do some things with this, but it's all right as just a static visualization as well. So we have our channels. We have our time series just over a course of, this is quite a long time. It's 250 seconds here. So can already see some weird stuff going on here, some boom, boom, boom. So some artifacts that will probably get dropped as we move through the processing analysis. What we can also do with the raw data is plot the power spectrum. Now I want to highlight this because this is something that came up in yesterday a lot, and also will come up this afternoon. The power spectrum from the EEG is a very information-rich and compact data type. Information-rich in the sense that you're capturing information across frequencies, okay? And compact in the sense that you're summarizing information over time. Okay, so when we're plotting the PSD here, like this is we'll just quickly check what the length of the raw is. 4,700 time points, and the sampling frequency is about 50 hertz. So we have 41,000 data points at 150 hertz sampling. So it's a lot of information there, and it's computing a power spectrum over this entire chunk. The key thing really is, what are we seeing in the power spectrum? Well, we're seeing pretty clearly the alpha peak. So I'm going to say alpha. This person has quite a slow alpha. You can see it in the EEG and the magnetometers and the gradiometers, and slightly less clearly in the magnetometers. But this person's principal rhythm is around about, I'd say, 7.5 hertz. So this is an interesting thing about EEG and MEG, right? We have these frequency band classifications, and typically as you have delta 0.5 to delta 2 to 4, theta 4 to 8, alpha 8 to 12, and then depending on how you define things, like low beta 12 to 20, high beta 20 to 30, and then gamma, low gamma 30 to 60, high gamma 60 to 60 plus hertz. Those would be a kind of standard set of numbers in terms of frequency ranges. So from that point of view, this person's principal frequency band is a theta, right? I said 4 to 8 hertz for theta. Now this points to something. Other people's peak frequencies will be typically more in the 8 to 12 hertz range. But certainly, like those boundaries, right? Like the border between low alpha and high theta. Like you shouldn't think of it as a categorical difference, right? It's like something that is clear in the data, like a principal oscillatory peak across multiple sensors. It's the same thing as if someone else has it 2 hertz higher. It's not a different thing. That's my hot take on it anyway. So we can plot it in a log scale as well, or like log on the x-axis, which gives us a little bit more, emphasizes a little bit more the 1/f, because it's already logged on the y-axis. So we see the 1/f kind of fairly clearly as this straight line here. And we see Frank was talking about FOOF analysis. So this alpha peak here, this would be your, FOOF Gaussian that you would fit on top of the straight 1/f background. Right, so now we're going to chunk this thing up. So typical thing that we do with EEG and MEG analysis, you do this thing called epoching. You do the same kind of thing with Fmri, but we have different terminology, and some things about how you kind of time things are quite different, because you have to be, you tend to kind of mark things a bit more sparsely with EEG and MEG, because your frequency, your temporal resolution is so much higher. You define events a little bit more clearly. So we kind of chop things up into epochs. Then we're going to do some stuff for those in a second, but firstly we're going to pull stuff from our parcellation. Now what we're doing here is, this should be familiar. It's different function, but it's pulling the same kind of thing. Labels here, it's actually an object with, so each of these things they have a certain type of information, but these are the location on a free surface average surface of each of these parcels. So the vertices that define the left bank of the superior temporal sulcus, left hemisphere, right hemisphere, etc. So these are our labels that we'll use for parcellating in a little bit. We're going to run our inverse solution. Now again, the inverse solution is where we say, okay, here's our channel level information. So ignore how we got the inverse operator for the time being, but we're going to use this to give us estimates of the source activity, and we're actually also getting, in addition, we're going to use our labels to define where we're going to try and pull the source information from. So this all gets pulled together into this thing called label T's here, label time series, and that is, again, it's a list. If I take a look at the first thing here, this is an array that is number of parcels by time. Okay. Now we can. Why don't we take a quick look at that? I also always stick stuff in pandas and kind of almost forgotten how to do native matplotlib plotting. Need to do a transpose first. It's over time. There we go. So that's our activity over time for this this epoch, because we have multiple epochs. We have. Look at the size of this, 55 epochs. Okay, so we have time series. Now, getting to our connectivity, what I'm doing first here is I'm doing something that's the same thing we did with the fMRI data. It was take the Pearson correlation. Now, I'm doing this for demonstrative reasons, because basically in EEG and MEG, functional connectivity with Pearson correlations is like naughty no-no. For an interesting reason, which is this problem that we call leakage, which is a bit of an unfortunate term, but it's source leakage. So basically, regions that are, well, estimates of activity that are in locations that are near to each other tend to be contaminated by each other, but they're contaminated what we call instantaneously. And the instantaneous part is because it's the propagation of the electrical or the electromagnetic field, right? So that actually propagates instantaneously. So we have this called like a zero lag contamination between nearby regions that's due to the physics rather than the physiology of neural interactions. And that is something that you see. It's kind of emphasized in the when you take Pearson correlations. So I'm showing you Pearson correlations here, and it's interesting that the structure of a Pearson correlation functional connectivity matrix is very much similar. Hopefully you see the similarity, very much similar to our diffusion connectivity matrix and our fMRI functional connectivity matrix. Left, interhemispheric block, right interhemispheric block. We have the secondary diagonals. But again, we have issues with a lot of this is probably due to genuine neural activity correlations, but we have this artifact that we kind of need to ignore. So what we typically do with EEG and MEG analysis is we move into what we call a non-instantaneous frequency, non-time instantaneous correlation patterns. We look at things like phase lagged synchrony, or we in this case what I'm showing you here is we filter in the we compute the Hilbert transform, and we find the instantaneous phase, and we look at phase-based correlations, which is what this function is doing for us here. And these are less prone to the leakage issue that I showed you just now. They're not completely invulnerable to it, but they're a little bit more sound. So we're just plotting these three things. It's actually basically three different versions of a phase-based functional connectivity metric, and they give you fairly similar results as well. So again, these are our connectivity matrices. We have a slightly different structure now in terms of the correlation patterns. We still do have a lot of emphasis, like weighting on the diagonals, but we have some block structure that seems to be principally driven by some regions are strongly correlating. So we're getting these kind of striped structures here, but we've lost a lot of the spatial structure that we saw here. And this is a typical thing, at least in my experience, with these kind of phase-based connectivity metrics with EEG and MEG, is they don't have so much of that canonical spatial structure in the connectivity matrix as you see in say diffusion and fMRI. So we're just going to do one final thing and then wrap up. Is just alternative plotting for each of these, but we'll just take a look at one, which is to arrange them on a circle. This is just a nice tool within M&E. Arrange them on a circle, left hemisphere and right hemisphere, and do a plot here. Now, this is, I would say this is a bit better. This is on the better side compared to the 3D brain plots that I showed before, and I was ragging on pretty hard. This is not so bad. I mean, you can see there's a sensible way of trying to do this kind of spatially structured network graph visualization. The number of nodes is not too crazy. We're not missing things because there's a third dimension that we're not really seeing properly. And we can see that there's strong left-right hemisphere correlations. That's coming through pretty clearly in this type of visualization. So this is actually quite a nice way of plotting your connectivity with these kind of circular plots. Yeah, okay. Nothing more to add on that really. So we have those for the 3 different connectivity metrics. They give us like somewhat different emphases. The cross-hemispheric connections are stronger in some than the others, but the basic structure is basically similar, as you can see between these 3. And that's also something I would say is kind of a feature of the phase-based connectivity that you metrics available, is they kind of tend to show similar patterns. And they also, interestingly, often show quite similar patterns across different frequencies. So you'll sometimes see kind of functional connectivity matrices plotted for different frequency bands, especially with another thing that we're not doing here called envelope correlations. And they're very similar between like theta, delta, alpha. So I think the field is still trying to figure out like what's the meaning of the fact that the correlation patterns seem to be quite similar across different frequencies. It seems to point to something that's not frequency specific, but that's kind of getting towards the methodological water cooler conversations there. Okay, guys, so that was our quick run through EEG and MEG to wrap up our Neuroimaging Connectomics session. We're a bit over time, which I apologize for, but we are done. And I'll just take a look at we have a question about Crowdcast. Will the links be there? Yes, the links will persist in Crowdcast, so you can come and kind of click on the links and follow what we were talking about.
Yeah.
Great.
Yeah, please remind you, once this session ends within, I think, 5 minutes, Crowdcast builds the video and puts it back at exactly this link. So, well, that doesn't work if you already left, but yeah, you can return to links from today or previous days if you needed to leave early, or not early, on time, while we were running late. Also, there's a question about what time we're going to start back, because we're a little bit late. Are we still trying to start back at 1? This is up to John. He's the one who has to talk for the rest of the day and eat.
Let's do 1:05, and
okay.
I'll be here at 1 for general chit chat.
Okay.
But we'll start proper at 1:05.
Cool.
Yeah. So thanks a lot, everyone, for your attention. That was Neuroimaging Connectomics. It was a roller coaster ride, but hopefully this was useful for you. See you guys at the next session.
We're putting up, and you have to eat, but I'll put up Gather Town in case people want to go chat. And don't need to bio break or eat.
Okay.
Cool.
Let's wait for the link to drop. A tour de force indeed.
Okay.
Okay. See you soon.